Today's lesson is all about, firstly, the genetic code, which is the sequence of bases in a molecule of DNA and how that gets translated into a protein, and then actually looking at the process of protein synthesis by extracting the information from a molecule of DNA and by a various number of steps, converting that into a string of amino acids in a particular sequence. So first of all, what is the genetic code? As I said just now, it's basically a sequence of bases on the molecule of DNA, and the exact order in which the bases appear on the molecule of DNA codes for a very specific sequence of amino acids. And as you will know, the primary structure of a protein is the order of amino acids that appear on that chain. And so you switch the code to produce a different sequence, you're converting it into a different protein. As you will remember from the previous lesson, what we might call the language of a gene has 4 letters representing the 4 bases. And remind yourselves what they are. A, T, C, and G, which represent the 4 bases: adenine, thymine, cytosine, and guanine. If we think about the code as being like a sentence where you have words and letters, the language of a protein has 20 words in it. And those 20 words are the 20 different amino acids that make up proteins. There are 20 naturally occurring amino acids in animals and plants, which are universal to all animals and all plants. There are a few artificial amino acids that are being generated in laboratories, but that is outside the scope of what we need to discuss. So, the question is, how do we code for 20 different amino acids using a sequence of 4 letters, A, T, C, and G? So, let's look at it in stages. If we said that one base, in other words, either adenine, thymine, guanine, or cytosine, represented one amino acid only, then obviously you could only represent 4 amino acids. Adenine would make one amino acid, thymine another, guanine another, and cytosine another. Obviously far short of what's actually needed. So let's go up one. What happens if we say 2 bases in a row code for amino acids? And if you put 2 bases coding for one amino acid, that would give 16 possible amino acids that could be coded for. You can work this out yourself by looking at these formulas. So there's the 4 possible bases. That's the number of amino acids that it's going to code for. That's the number of bases that you're going to use, in this case, 1 base. And that gives you only 4 amino acids. If we take 2 bases, which could code for an amino acid, so there's the 2, there's the 4 bases, 4 to the power of 2 would give you 16. So that would give you only 16 amino acids, which again is far too few. We need at least 20. So finally, if we look at 3 bases coding for one amino acid, there they are. Remember, the 4 represents the 4 bases. So this gives a number of amino acids that are possible is 4 to the power of 3, which is 64. Far more than we need. But that's actually quite useful because most amino acids are actually coded for by more than one sequence of bases. And we'll come on to that later. So this is the most efficient way. If you thought of making the code for an amino acid 4 bases in a row, so 4 to the power of 4, you can work out what that will be. That's even more inefficient We need 20, that's not enough, that's too many, so that's the best we can do. And so biologists have concluded that the number of bases on a sequence of DNA that is going to code for one amino acid is 3. And so for that reason, the genetic code is referred to as a triplet code. Triplet 3. So, in summary, 20 amino acids to be coded for, 64 base triplets to use to code them. Fortunately, you never have to learn the sequences of bases that code for particular amino acids, but you do need to have an understanding of how it works, and periodically a question will come up relating to the coding of bases for a particular amino acid. If you go to page 513 in your books now, so pause the video if you need to go and hunt for your book, and we'll just have a look at one or two things. So page 513, the chart at the top of page 513, which is headed Appendix 2, DNA and RNA triplet codes. We're interested in the DNA only. The way the code works is that you look for the amino acid that you're after. Let's say, for example, we're going to be looking at valine, which is VAL, which you find at the bottom of the first column of amino acids. The way you read the codes that will make valine is you start off with the letter in the first position, which is on the left-hand side of the chart, and in this case it's a C, standing for cytosine. The second position, which runs along the top of the chart, will be A, standing for adenine. And then the third position, down the right-hand column, could be, in this particular case for valine, be either adenine, guanine, thymine, or cytosine, because you've got there A, G, T, and C all lining up with VAL for valine. So the genetic code for valine is either CAA or CAG or CAT or CAC. And any of those 4 sequences of bases will ultimately, at the end of the protein synthesis section that we're going to be looking at shortly, result in the production of a molecule or the incorporation of a molecule of valine in a particular protein. We'll come across this chart again. I said just now that each amino acid has more than one code word. We just looked at the example of valine. And the phrase which defines that, when it's not one specific sequence of 3 bases, is that the code is degenerate. And degenerate in this case simply means there are more than one possible sequence of bases to code for any particular amino acid. It's also non-overlapping. So there, in the middle of the page here, is a sequence of bases in a molecule of DNA. Remember, this is the steps in the ladder of the molecule of DNA. So we've got adenine, thymine, cytosine, guanine, adenine, guanine, and so on. And you break that up into triplet codons. So ATT, there it is, CGA, there, GGC there, and GGT at the end. So here we've got a sequence of 4 triplet codes out of the whole length of a molecule of DNA. And during the process of protein synthesis, this sequence is read all the way along a molecule of DNA and is converted eventually into a sequence of amino acids. If you like, you might like to spend a few minutes looking at what these 4 sequences of bases represent by looking back at the chart we were looking at just now. So you can work out what these 4 amino acids would be. So in summary, the genetic code is a triplet code because you need at least 3 bases. It is degenerate because there are many codes for 1 amino acid. It's non-overlapping, so 3 bases code for only 1 amino acid. You can't then start the next sequence earlier. For example, there's a sequence. That's how it works. You can't make it overlap and have those 3, and then those 3, and then those 3, because you've got overlapping letters. So they are distinct triplet codons. And the last point on the board here is that it's universal. This code works for every single living organism that contains DNA. Whether it's a bacteria, whether it's an oak tree, whatever it is, every single living thing is universal in the way that it codes. So the code for an amino acid in a plant is the same code that it would be in an animal. Here's a molecule of insulin. What I would like you to do as an exercise to go with the prep that you will find on a different document is I would like you to draw the first 6 amino acids there, and beside each one write the triplet code that represents it. So what's the triplet code for phenylalanine? What's the triplet code for valine? And so on. So you can do that as part of this week's prep work, using the chart at the back of the textbook. So now let's move on to protein synthesis. There are 2 major processes. Transcription is where the message, if you like, or the sequence of base codings is extracted from the molecule of DNA. The translation is how that code is then translated from a sequence of bases into a sequence of amino acid that will form the primary structure of a protein. So we'll start off by looking at transcription. Remember that this happens inside the nucleus. Transcription is the first process. The DNA is on the chromosomes inside the nucleus, and so therefore the transcription of the message, or the copying of the message, has to happen inside the nucleus. And it's copied by creating a molecule of messenger RNA, sometimes as shown there, written as lowercase m RNA. There are a couple of differences to remind yourself about the differences between RNA and DNA. First of all, RNA is a single strand rather than a double helix. So it's a single strand of sugar phosphate, sugar phosphate, sugar phosphate, each of which has a base attached to it. The second difference is that thymine is replaced by uracil. You can check that up in an earlier part of the chapter. There's a short section on RNA in that particular section. So you don't find thymine in a molecule of RNA, but you do find instead uracil. You still find adenine, guanine and cytosine, but joined by uracil rather than by thymine. And the third difference is that the sugar is ribose rather than deoxyribose. And again, you could find the difference. You don't need to know the structural difference between ribose and deoxyribose, but you can find it if you look back at the work we did last week at the bottom of page 112. There's a molecule of ribose and a molecule of deoxyribose, and the only difference between the two is the fact that on carbon number 2 there's a hydrogen ion instead of a hydroxyl group. The rest of the molecule is exactly the same, but you can check that up for yourself. So, the process. Here is a molecule of DNA, a section of a molecule of DNA, with the usual sequence of bases being paired, adenine with thymine, cytosine with guanine, and so on. The first thing that happens is the molecule unzips, because, as you remember, there are hydrogen bonds between these sections here, and the hydrogen bonds are broken using an enzyme reaction, and then you end up separating the two nucleotide molecules. There they go. One of the strands of the DNA molecule is referred to as the sense strand. That actually contains the code itself. So the code that we're reading here is TCGA, not that one. This is the sense strand. And what happens is that free molecules of nucleotide, of RNA nucleotide, so ribose and a base and a phosphate are floating around in the nucleus, and they pair up to each of those. There they go. So ribose as opposed to deoxyribose, uracil as opposed to thymine. So this was thymine in the DNA molecule, it's replaced by uracil. But the rest of it's the same, you see. There was a cytosine in the DNA there, cytosine in the RNA here, guanine, adenine, and so on. These form hydrogen bonds to join them together. And then you end up with these being joined together by hydrogen bonds. So there's a bond there, a sugar phosphate bond between this base, and so on. So it's basically forming a molecule of RNA attached at this moment in time to the molecule of DNA. This is now referred to as messenger RNA because it's carrying this message from the nucleus to another part of the cell where protein synthesis is going to take place. Off it goes. Having produced the molecule of messenger RNA, the two DNA strands join back together again. So the DNA isn't destroyed, it's brought back together again. And it can be ready then for use by the next time you need to produce a molecule of protein. So the DNA remains unaltered at the end of the process. And then finally the DNA molecule winds back up into a helix. So that's basically the process of transcription. New word. The sequence of 3 bases on the messenger RNA coding for amino acids are called codons. Okay, so the base triplet on the DNA has now been converted into a codon of complementary bases on the molecule of messenger RNA. Something that we'll talk about much later on in the syllabus is the fact that there are certain parts of DNA that are responsible for coding for things, but not for proteins, not making directly proteins. So there are sections of the molecule of DNA that get in the way, if you like. And so they have to be snipped out of the messenger RNA before it's converted into protein. The Bits that are not used for making protein are referred to as introns. So you think of a molecule of messenger RNA as being a sequence of bases that is copied from the DNA which will make a protein. Then there's a section which doesn't make proteins. Then there's another section that does, and so on. And those things that do not make proteins, the introns, are chopped out by enzymes, and here are the sequences of bases that make the proteins in the exons. So it's a waste to replicate and try and produce things from the introns. And so before the messenger RNA leaves the nucleus, it's snipped up by enzymes. The introns get taken out. Enzymes then come along, which then will ultimately join those exons back together again. Like so. So now we've got a workable code which is designed exactly to make proteins. None of this irrelevant stuff has been left in it. So, here's the nucleus. So we've got to the end of transcription here. Here's the molecule of messenger RNA that's been produced by the process of transcription from the DNA molecule inside the nucleus. Proteins are actually made in the cytoplasm, and there are a number of things necessary for that. So the first thing that's got to happen is that the messenger RNA has actually got to come out of the nucleus, and it joins on to a number of ribosomes. You'll remember that ribosomes are the smallest of the organelles, and they come in two components. And in a eukaryotic cell, these are referred to as ATS ribosomes, and in prokaryotic cells, they are 70S, a little bit smaller. They join on to a particular point on the molecule of messenger RNA in order for the process of translation to begin. So, we've come out of the nucleus, we've finished with transcription. Here's the messenger RNA joined on to a ribosome, and that is now ready to begin the process of protein synthesis. So, we're now looking at translation. Here's our molecule of messenger RNA with the base triplets, which are called codons. 1, 2, 3, 4, 5, 6, 7, 8 codons. The other thing that's necessary for this process is another type of RNA which is referred to as transfer RNA. It's a funny shaped molecule. It's not a single strand all over. Basically what a molecule of transfer RNA is it's basically something that looks a little bit like this. It's got 3 bases sticking out from the bottom, and remembering that these are referred to as codons, this is an anticodon. And this is the strand of transfer RNA, and it does form bonds, various places to hold it together into the shape. And at the other end, you have an amino acid. And the thing about this is that there is a specific amino acid that will attach to a particular molecule of transfer RNA dependent on the bases on the anticodon. This is shown in more detail in the textbook. So, the sequence of events. In a molecule of a section of the messenger RNA, there are things called start codons and stop codons. This enables the mechanisms of the cell to know which is the first amino acid that's going to be made. So in this case, AUG is a start codon, and UGA, the other end, is a stop codon. And the 2, 4, 6, 8 codons here each of which are going to code for a particular amino acid. So you're going to end up with 8 different amino acids joining together. So, I think you can perhaps see what's going to happen first. You're going to get the complementary base pairing. Adenine will pair with uracil. Remember, we're talking RNA, so there's no thymine. Uracil will pair with adenine. Guanine will pair with cytosine. So that's the first step. And what this process has done is fix an amino acid in place in the cytoplasm, on the rough endoplasmic reticulum in the case of eukaryotes. So this is set in a position. Here we've got another molecule of transfer RNA with a different amino acid attached to it. Remember, each amino acid has a specific sequence of bases on the anticodon. So once again, you can see what's going to happen here. This particular codon is made of 3 cytosines. And so this will attach to it because these are the complementary bases, guanine and cytosine. So we've now got 2 amino acids close together. What are we going to do to join those together? What sort of bond are we going to make to join the 2 amino acids together? There it is. A peptide bond, which I hope you remembered. So we've now made a dipeptide, the first 2 molecules of a protein. So now we seem to have filled up the ribosome. There's no spaces for any more transfer RNA to join on. So the next thing that happens is that the first one disappears, and that will be recycled. That will join on to another amino acid and can be reused. The ribosome then shunts along 1 codon from right to left, exposing this next set of bases in the codon. So there we are, another amino acid, different one, with the anticodon cytosine, 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 which is going to join nicely onto the 3 guanines there. Once again, a peptide bond, and then the whole thing continues. And eventually you end up with a sequence of 2, 4, 6, 8 amino acids, 1, 5, 6, 7, 8, relating to the 8 bases, the 8 codons that were on the messenger RNA molecule. So what we've done here is we have created a protein, the primary structure, with a sequence of particular amino acids. And the sequence is exactly coded for by the sequence on the messenger RNA, which is in turn coded for by the original sequence of bases on the molecule of DNA. It's important for the cell to know where to stop. A protein is only a certain number of amino acids in length, so when it reaches the stop codon, at that point the process stops. This is not one thing, it doesn't happen just once. You can picture in real life a number of ribosomes following each other along the sequence here, churning out chains of amino acids. So it's not just one ribosome trundling all the way down and then another one trundling all the way down. It's a whole sequence. So you're producing lots of molecules of protein at the same time as each of the ribosomes follow each other along. So finally, on this topic, we need just to look briefly at what's referred to as a gene mutation. We'll talk about it in a bit more detail in other topics. But a gene mutation is where something goes wrong with the process of transcription or translation. Remember that we've already looked at one problem with mutations, and that is in the replication of the molecules of DNA during interphase. It is possible for a mistake to be made and something to go wrong, but it's a tiny, tiny probability. Here we've got several things taking place. We've got making a molecule of messenger RNA from the DNA, and then we've got making a molecule of protein from the messenger RNA. So there are more opportunities for errors. And when errors happen, they are called gene mutations, when basically what happens is something goes wrong with the sequence of bases that is being transcribed or translated. So a gene mutation is a change in the base sequence of a gene. And of course, if you change the sequence of the mutations in the gene, you're going to change the sequence of amino acids in the final protein. So if there's a mistake in the gene, there's going to be a mistake in the protein at the other end of the process. So for example, here is an example of a simple mutation. There is a codon, GGG, and that happens to code for the amino acid proline. If you change one base, in this case, the first one, changing from guanine to cytosine, now you've got CGG, and now instead of coding for proline, it codes for alanine. You can check that up again in the appendix at the back of the textbook. This is referred to as a substitution mutation, where you simply change one base for another. But it does have an impact. We've talked briefly already in an earlier topic about hemoglobin and how, if there's a mutation on hemoglobin, changing one amino acid creates an issue. It creates a change in the structure of the hemoglobin. Here is the example that we looked at. A gene mutation, sickle cell anemia, so we're looking at hemoglobin. Thymine is substituted for adenine, so a particular triplet in the gene for hemoglobin codes for the amino acid valine rather than the correct amino acid, which is glutamic acid. Okay, so by changing one base, thymine into adenine, you are then changing the amino acid coding from glutamic acid, which it should be, to valine, which it shouldn't be. And you might remember that the problem with valine is that it has insoluble or hydrophobic R groups, which means that the molecule of hemoglobin is less soluble than it should be. And what are the implications of that? There's what I said just now. Valine has a non-polar R group, whereas glutamic acid is polar, creating a hydrophobic spot on the hemoglobin, so it makes it less soluble. That causes more molecules of hemoglobin to stick to it, so hemoglobin is no longer a nice molecule floating around in solution, carrying out its task of oxygen transport. It's tangled together with other molecules, which then stick together in long fibres, which are quite rigid. That distorts the shape of the red blood cell, and it also makes it less efficient at carrying oxygen. And those are some of the problems. The red blood cell becomes distorted, sickle-shaped, gets stuck in capillaries, so it can lead to blood clots, because remember, a red blood cell is normally very pliable, it's flexible. Normal red blood cells don't move around the body as fast as they should do, and so the person who has sickle cell is anemic and is unable to carry sufficient oxygen to the tissues. And that basically concludes the lesson on protein synthesis and the genetic code.